Okay, our next presenter is Paul Lakin from ViaCyte. Paul? Okay, thank you. And I'd like to thank the presenters for giving me an opportunity to tell you about ViaCyte, a company formerly known to some of you as Novacell. ViaCyte is a leading company in the emerging field of regenerative medicine. It's a privately held venture-backed company. Our principal site of operations are here in San Diego, as well as in a small satellite facility in Athens, Georgia. We have about 53 full-time employees at the company, and we are really focused on a single, what we believe will be disruptive product for diabetes. This product combines a pancreatic beta cell precursor that was derived from a human embryonic stem cell line. We encapsulate that in an immunoprotective device, and we believe that this together has the potential to transform the way patients with diabetes are treated. We believe it represents a potential, essentially a cure for type 1 diabetes and an important therapy for insulin-dependent type 2 patients. The company has been working at this for quite some time now, and we are now approaching clinical testing. I recently had a pre-IND meeting with the FDA, and we have been generously supported by CIRM and by JDRF. So these are the core elements of our product. It starts with a renewable cell source. It's a human embryonic stem cell line, as I said, designated as Site 49. We then take that and differentiate it through a multi-step process to a pancreatic progenitor with beta cell potential. Essentially, this is a prodrug concept, because we then implant this into the patient in a durable immunoprotective device. We have a proprietary device we call the Encaptor Drug Delivery System. And once this is implanted in the patient, then over a several-month period, the cells continue to differentiate and mature and form insulin-producing and other factor-producing cells. So a lot of work has gone on over the last few years in really getting this into a very reproducible, a regulatory compliant format that we can scale up for clinical trials and eventually commercialization. So we have these, the cell line, the human embryonic stem cell line that we started it with is ethically derived under GMP compliant conditions. We bank this, we have several banks in several locations that gives us essentially an unlimited renewable source of these cells. They have been extensively tested for advantageous agents. They show karyotypic and phenotypic stability. We use xeno-free culture. And importantly, we can rapidly expand these from 10 million to billions of cells in only a couple weeks. The next big part of this is the differentiation protocol, which most of you in here are pretty familiar with, the whole concept of this. But it's been worked out to be entirely reproducible. This has been done to be regulatory compliant with steps along the way as we do this. It's a four-stage or four-step differentiation process that takes about 2 weeks. What the end stage of this is get to these pancreatic precursor cells. As you can see over here, when we take these and we implant them into the animal, this is with or without the device, they then mature over a several-month period to make these insulins, somatostatin, glucagon-producing cells. What's shown here is a typical human islet after development in an animal for about 360 days, and here's where we put our product, which we label PEC-01, into an animal for 377 days, and you can see very comparable in terms of the production of these various hormones. The next important part of this is to manufacture this to the level that we need for clinical trials and eventually commercialization. Our initial manufacturing is really scaled for those clinical trials. And again, it involves expanding these human embryonic stem cells, differentiating them over that 2-week period. We're then able to freeze the product, PEC-01, and therefore be able to do all the stability and various other assays on it. And then when it's ready to go to the clinic, we thaw an aliquot of the cells and then we load them into a device, and then that is shipped to the clinic. So what the clinic gets is the device loaded with the PEC01, these pancreatic precursor cells. The device itself that we use to encapsulate these cells to increase safety and eliminate the biodistribution is a biocompatible, biostable device. It's a proprietary device that we developed. It facilitates vascularization. Importantly, it excludes the host cells and retains the grafted cells. It freely allows the diffusion of oxygen, nutrients, proteins, including, of course, insulin, which are freely transported across the membrane. It protects these allogeneic cells from the host alloimmunity, and we believe it should also protect against the autoimmunity that's seen in type 1 diabetes, and this is based on other cells that have been used in similar devices. Importantly, this device, as you'll see in the next slide, we put in subcutaneously, and it's removable. And that has some really interesting safety as well as efficacy implications. So now we have the cells manufactured, we have the device. This is showing here what the device looks like. It's kind of a cartoon of it, but this would be a little bit different when we get to the commercial or the human device. But it basically has a fairly thin lumen that's loaded with cells that facilitates oxygen diffusion and all of the factors coming in and out that are necessary for the regulation of these essentially beta cells. The device is implanted subcutaneously. Because once it's implanted, it takes 2 to 3 months to produce, to mature and start producing the insulin. We have to implant it in somewhere where there won't be a needle stick, because of course these patients initially, when we first implant this device, will still be using insulin, these type 1 diabetic patients, and so they will still be injecting, so we want to keep away from the possibility of a needle stick into the device, but there is a lot of real estate that we can use for doing this. This shows from a mouse now, where we do these studies in a SCID-beige mouse model. This is a device that has been implanted with the PEC01 cells and has gone for 18 weeks in the mouse and then is removed. And this is characteristic of what we see. We've done literally thousands of animals now with this product. And what we see is we see a thin encapsulation of the device, as you can see along here. But we see tremendous vascularization. And the mesh that you're seeing here on the device is a mesh that is put over the diffusible membrane. The mesh provides protection and structural integrity to the device. But what you can see is these vessels, as they grow in over that 18-week period, are completely covering the device and going in and out of this mesh, and supplying the oxygen and glucose and other factors that are necessary for the growth of the cells. Because the other thing, the characteristic that we see is that even though we have this tremendous vascularization, we see it have only a few feeder vessels, which makes, again, this something that can be easily removed with very little trauma to the patient. So this shows a device now that's been implanted 16 weeks in a SCID-beige mouse and then taken out, and then we section it and we look at what we find in it. And again, you can see this tremendous vascularization. These are all blood vessels along the walls of this. These, by the way, are artifacts from this mesh that is protective of the device. Over the top of the device, this is where the mesh goes in. This is the actual membrane. These are the cells that we put in them that have now matured into insulin-producing, essentially human islets. And so we have this tremendous vascularization, and then if you visualize this, looking at staining for insulin, glucagon, and somatostatin, you can see a very robust production of the various of insulin and other factors. And this also shows down here, we look at C-peptide now. We're measuring human C-peptide production in this mouse model. And this is after a glucose challenge. After 30 and 60 minutes, you can see the C-peptide comes up very rapidly and then begins decreasing as the glucose is normalized, just as you expect. This now is just a representative study. Again, as I said, we've done literally thousands of animals with this device drug combination. This is one representative study. It's looking out at a group of animals, again, a mouse study where the device is implanted and allowed to mature. And then 7 months after this device is implanted, the animals are given STZ to essentially destroy their endogenous beta cells, the mouse beta cells, but leaves intact the beta cells that are in our device. And what you can see here is that all the way out here, when we get to the STZ, we have very well controlled, very similar. The red is actually a fat pad insertion of cells, so it's outside of the device. But the rest we have in the device. And when we continue this out, we see a good maintenance of blood glucose. And then after 70 days, we remove the devices. And what you see is immediately the animals, once the device is removed, become hyperglycemic. So a very nice demonstration of the ability of this. Interestingly, the other thing we see consistently in these mouse models, the typical level of glucose in a mouse is about 130 to 200, which for a human would be considered hyperglycemic. Generally in the mouse model, we see that once we put the device in with the human cells, they move down near the human set point of about 100 to 110 milligrams per deciliter. Another important aspect of this approach is that we have never seen any evidence for any potential for hyperglycemia. In fact, in these mouse models, when we put this device in, it's a very small device designed for the mouse, but if you put that same size device in a human, it would be like putting a cafeteria tray on your back. So these mice are very much overdosed even with this small device drug cell combination, about 10 to 30-fold overdose. And here what we're showing is, again, in this mouse model now, we do a glucose challenge. So we take these mice, we give them a large bolus of glucose, and as expected, the blood glucose rises, but fairly rapidly the insulin is released, and we now see a reduction at glucose back to normal levels. But even though this is 10 to 30-fold overdose, we see no hypoglycemic overshoot. So we think this is a big advantage for this approach. So as I mentioned at the beginning, we are on the path to the clinic. We've been working very hard to get this into a scalable format and into a regulatory compliant format. We recently, in the last couple months, had a pre-IND meeting with the FDA that went very successfully. At that meeting, we laid out the IND enabling studies that would be required, as well as the initial protocol. And so we're on track initiating those IND enabling studies right now. We expect to follow the IND around the end of 2013 or early 2014 and initiate clinical trials directly after that. The initial trial design is going to be a phase 1-2 study. We're going to go directly into patients with type 1 diabetes. And we will be looking for safety, but also for early proof of activity and mechanism. Even in the first patient cohort, the way we're going to do this, we will have some sentinel devices in there that we can pull out at different times, and we can look and see if the cells are differentiating as we expect without the need for any immune suppression as we expect. So even in the very first cohort, we should start seeing evidence for proof of mechanism. And by the third cohort, we're planning to provide enough of the product to essentially render the patients independent of insulin. CIRM and JDRF, especially CIRM, have been early and enthusiastic supporters of our efforts. We received from CIRM around $26 million to date, and we just last week were the beneficiaries of an additional grant from CIRM for $10 million. I loved when we read the reviewers' comments that CIRM posted to their website, The reviewers called VCO1 our combination product, the holy grail of diabetes treatments. That figures prominently in all my presentations now. On the IP part of this, the company has been around for about 10 years, so this has been kind of an endogenous, if you will, project. So a lot of the IP has come from internal development and discovery, and we have patents and IP protection all along the way. Final slide to summarize. Our product has the potential to address what we believe is a major unmet need in patients by providing islets from pancreatic precursors, which are self-regulating very much like the human pancreas. We believe we'll get superior glycemic control, no needle sticks, no glucose monitoring, no risk of hypoglycemia. It's a subcutaneous implant. It's a simple outpatient procedure, low risk of complications. It can be easily removed and replaced if needed. One of the concerns on this type of approach is with keratoma formation. If there is one, it will be contained in the device. We've never seen that in several thousand animal models. And we expect it will lead to a higher quality of life with no limitations on physical activity. And obviously the market potential for this is very high. We hope to reduce the medical and indirect costs of type 1 diabetes, which is about $15 billion per year in the US alone. And mainly I've been focusing on type 1 diabetics, but obviously we believe this also would have a role in the insulin requiring type 2 diabetics.